Richard is a second-year PhD student in my lab and in chemical and biomolecular engineering. Coincidentally, he also did his bachelor's degree at UC Berkeley, and I'm guessing you might have, Afiksha and Richard, you might have overlapped a little bit, but anyways, so Richard has worked in undergrad on analysis and synthesis of natural products from microorganisms. And now he's continuing to develop analytical techniques and perfecting microbiology skills to study microbial metabolism. And so he's going to present one of his projects on glycolysis. And yeah, so Richard, the floor is yours.
All right, thank you, June, for the introduction. And thank you, Apeksha, for the great talk on the first half. Obligatory go Bears, but also go Bruins, everyone here. So yeah, hi, as June introduced me, my name is Richard Law. I'm a second-year graduate student in his lab. And today I'm going to talk about one of my projects, which is about studying how parallel glycolytic fluxes redistribute in dynamic nutrient environments. And hopefully from this talk, we can give you guys a bit of a taste of the flavors of the different ways that we at the Park Lab study biology. And specifically at the Park Lab, we study biology under the lens of metabolism. And this is because while we often think of the central dogma of molecular biology to talk about the kind of processing of genomic information from DNA to RNA to the proteome, to capture certain aspects of biological phenotypes, it's often also beneficial to kind of extend that pipeline to talk about metabolites and metabolism. Metabolism is just the interplay of the many metabolic reactions and biochemical reactions that are facilitated by those enzymes coded by the proteome. When you want to look specifically into the actual rates of these reactions rather than just the concentrations of those substrates and products, we're talking about fluxomics data. Fluxes are basically akin to the rates of passage of individual metabolites through metabolic networks. You can think of them basically as the rates of biochemical reactions. And by studying fluxes, we hope to offer insights into metabolism in action and help address a bunch of different questions from medicine, nutrition, disease, and also synthetic biology. And regardless of kind of which field of biology you're interested in, I think there's one key interesting query about how metabolism is designed. And regardless of which organism you look at, many model organisms possess what I would consider to be called redundant or parallel metabolic pathways. If you could think of different biochemical reactions and metabolic pathways to serve functions for cell life and growth and proliferation, you could map out these individual network of reactions and categorize whether they're essential to cellular growth and survival or conditionally essential depending on the growth conditions that you're putting your cells within. And then in many cases, whether these pathways, genes, or regulatory features are actually redundant to the cell. And this is actually a feature characteristic of many different organisms. I'm just showing this model that previous researchers have done on E. coli metabolic models and Baker's yeast metabolic models. And so, for example, in the case of E. coli, up to around 37%, at least in this model, of metabolic pathways and cellular metabolic networks are considered to be redundant inside the system. And among those redundant pathways, some of them are deemed to be considered non-efficient or unoptimal in terms of their operation or expression, but they're still actively utilized in different parts of cellular metabolism. So this kind of begs a very fundamental question about how metabolism has evolved and is designed. Specifically, we're asking, why do organisms retain these redundant and parallel pathways? And specifically in situations where cells finally do possess these redundant or parallel pathways, how are these redundancies evolutionary competitive in the sense of proliferating accelerated growth or competition against other competitive similar organisms? And this is an interesting question that's not just relevant to niche corners of the metabolic space, but actually in central carbon metabolism and primary metabolism as well. I think one of the key metabolic pathways that everyone learns in their sophomore level chemical biology course is glycolysis. And that's because glycolysis is often considered to be a very exemplar and essential pathway across a wide conservation of different organisms. Glycolysis is highly conserved across almost all eukaryotes, and many prokaryotes possess glycolysis as their source of carbon intake. Now, I don't want to give too much of like a sophomore biochemistry lesson here, but in brief, glycolysis is just the conversion of carbohydrates as a carbon source from glucose into 2 units of pyruvate. And in doing so, this allows cells to process outside carbon-based nutrients for a wide variety of functions, whether that's for the conversion of nutrients into biomolecular building blocks, or for just bioenergetics by processing pyruvate through the TCA cycle and other routes in the metabolics pathway. Glycolysis has also been shown in previous studies to be highly evolved and regulated so that it can rapidly adapt its fluxes or metabolic rates in response to intracellular and environmental demands. And so this kind of map of the metabolic pathway of glycolysis, we'd assume to be an exemplar of optimal functionality and is kind of like the keystone pathway that many cells would use. That being said, this is only one half of the story of glycolysis, or even 1/3 per se. Because while the glycolysis that I described just now is really just considered to be the Embden-Meyerhof-Parnas pathway, or EMP pathway, as I'll call it for the rest of this presentation, but in many prokaryotes, there exists a parallel glycolytic pathway called the Entner-Doudoroff, or ED pathway. Now, these pathways are carbon parallel in the way that they both begin with the same carbon substrates of glucose and end in with 2 molecules of pyruvate. However, stoichiometrically, they differ from their bioenergetic yields. Specifically, 1 molecule of glucose will yield 2 ATP inside the EMP pathway, but notably in the ED pathway, only 1 molecule of ATP is generated per molecule of glucose. Just stoichiometrically, this makes it seem that this existing pathway, the ED pathway, is much bioenergetically less efficient than the EMP pathway and would, in that sense, be unfavorable for optimal use in a highly evolved organism. However, phylogenetic studies of different heterotrophic prokaryotes have demonstrated and shown that the ED pathway is, in some cases, exclusively used. And interestingly, for the topic of this research presentation, in organisms such as E. coli, which are a very highly studied model organism for metabolism, E. coli sits inside this purple region of 14% of organisms that actually possess both the EMP and the ED pathway. So rerouting that kind of fundamental question of why would organisms possess, for example, parallel metabolic pathways, we're kind of honing into the tune of specifically with the route of glycolysis to our research statement of what is the role of the ED pathway in some evolved organisms, such as E. coli, that already possess a more glycolytically efficient pathway, such as EMP glycolysis. And to conduct this investigation, I'm going to kind of go through 3 flavors of kind of studies that we went through. First, through some computational modeling and estimates of growth rates and growth phenotypes across cells, just by using computational models paired with our experimental measurements. Then after those measurements, we're going to kind of look into some more classic microbial cell growth experiments and metabolomic techniques to kind of elucidate a surface level insight on what might be going on in the ED pathway in these organisms in specific growth conditions. And then afterwards, we're going to be using isotope tracers and developing quantitative flux analysis tools to kind of actually assign numerical values to these ED pathway and EMP pathway fluxes. And hopefully by the end of this, these 3 different investigations, we can come to some sort of conclusion that stimulates that the ED pathway enables a rapid upshift of metabolism during sudden nutrient availability. So going off into kind of part 1 of this step, we started our investigation by conducting just a flux balance analysis of cellular fluxes at steady state. So flux balance analysis is a computational and modeling technique used by metabolomic researchers to kind of estimate fluxes based off the balances of steady state. To conduct flux balance analysis, you begin with genome scale reconstructions of the metabolic networks. In doing so, you can generate what we call the stoichiometric or S matrix, which basically just lists out all of the metabolic reactions possible and the metabolites that are consumed and produced. From that genome scale model, you can then employ a couple of constraints such as mass balances, which dictates that at steady state growth, the product of all of the stoichiometric matrix by all of the reaction fluxes in the system should equal 0, which corresponds to no accumulation or deaccumulation of metabolites in a steady state system. We can additionally constrain individual fluxes within the flux vector based off of our knowledge of real world biology or just by specific experimental conditions that we're hoping to explore inside this mathematical model. And then finally, we define an objective function, which is just a linear combination of these different fluxes that our constraint-based analysis will try to optimize. And once you input all of these variables, we can find a solution that's obtained via linear optimization to kind of determine some vector of simulated fluxes that best match the system that we're modeling. So we first conducted this flux balance analysis, or FBA, using a genome scale model of a K12 strain of E. coli with specific constraints to kind of map out glycolytic growth using a glucose intake rate similar to our experimental values. And also we tested out what would happen to the model of growth, which would be our objective function in this situation, in the case of knocking out specific genes along the glycolytic pathways. And in doing so, we just constrain our fluxes to 0 millimoles per hour per gram cell weight. And what you can see on the right is that while some of the key steps in the EMP glycolysis, knocking out those steps has an effect on the cellular growth rate inside the FBA model of steady state growth, knocking out of the ED pathway enzymes, which are delta EDD and EDA on the far right, have little to no effect on steady state growth rates. Now, these mathematical and computational estimates do reflect what we see inside experimentally when we just do individual growth curves. And so this initially just kind of paints out a picture that, oh, at steady state growth, when cells are optimized to utilize their metabolic pathways for the highest amount of carbon and bioenergetic yields, it seems that the ED pathway plays very little or low importance. But then this begs us to ask the question, if it's not in steady state conditions, could the ED pathway have relevance in transient dynamic conditions? And so we set up 2 experimental conditions to kind of study whether nutrient upshift plays a role in the usage of the ED pathway inside these cells with parallel glycolytic routes. Glycolysis can be regulated in many different ways. And so we designed 2 separate experiments to modulate glycolytic upshift on 2 different kind of fundamental mechanisms. On the left, we begin cells growing in the absence of glucose. Instead, they use acetic acid or acetate to kind of input their carbon sources through gluconeogenesis. Then by adding glucose, cells will preferentially use glucose instead of acetate and then upshift their glycolytic fluxes. Nitrogen metabolism is also highly interconnected with carbon metabolism. And so low nitrogen conditions can also inhibit the rate of glucose import into cells. So on the right panel, we conducted an experiment where we had a situation of first growing cells in a nutrient low environment with low amounts of nitrogen before upshifting their ammonia concentrations inside liquid cell cultures to rapidly upshift glycolytic fluxes. And then we hope to see if there are any biological differences in phenotypes of growth and whether that could play a role in the relevance of the ED pathway. So we looked at these growth curve experiments specifically from glucose limited to glucose upshifted and glucose repeat growth. And what we notice is that while similar to our FBA estimations, the steady state values of growth are largely non-significantly different from each other. These 2 different strains, whether it's the ED pathway knockout or a wild type strain, which has both the EMP and ED pathways, they actually demonstrate different profiles of how they increase their growth rate over time. This is similar for the case of both glucose upshift and also a nitrogen upshift, where we have a unique change of growth phase. And so to kind of more quantifiably measure this upshift growth conditions, we quantified just roughly what we termed to call growth acceleration, which is just the time derivative of the growth rates between that region of transitory growth phases. And what you can kind of biologically assume this growth acceleration to be is just how quickly can cells adapt to a sudden change in their nutrient environment, and how does that change kind of reflect the metabolism of the cell. And so we quantify these differences, and we notice there to be significant differences in different phases of upshift, depending on whether or not cells possess the ED or EMP pathway. And so this obviously kind of invites us to conduct more explorations on what's happening inside cellular metabolism. To get better insight into maybe the actual metabolic profiles of what's happening inside these 2 ED pathway, this ED pathway knockout versus the wild type, we conducted metabolomic experiments, which uses liquid chromatography mass spectrometry to take intracellular extracts of metabolites in the cell and profile them over time. And in doing so, we can generate these maps of metabolite profiles over time and kind of understand if the metabolome is changing in response to this nutrient upshift, and if that could be implying or related to changes in fluxes. So I'm not going to inundate you with kind of like metabolomic profiles of the entire central carbon metabolism, but instead I wanted to focus mainly on just some of the main glycolytic intermediates that exist in both the EMP and ED pathway. And so while the profiles are relatively similar in the kind of the range of what happens from our understanding of changes from, say, gluconeogenic to glucose growth, for example, P levels drop significantly, or in the cases of rapid upshift of glycolysis from ammonia upshift, which has a rapid increase in pyruvate levels. One notable result from this metabolomic studies I wanted to point out is actually a dynamic change in KDPG levels. KDPG, if it wasn't clear before, is a unique glycolytic intermediate that is exclusive to the ED pathway. And so in the wild type strains, which actually possess both EMP and ED pathways, we notice that within the first 3 to 5 to 10 minutes, instead of rapidly upshifting their glycolytic profiles, which would be attuned to just kind of a general proportional increase of glycolytic fluxes from both the EMP and ED pathways, we see there to be an initial decrease of KDPG intermediates within those first 3 to 5 minutes. And so basically the takeaway here is that these metabolomic profiles are implying some amount of ED pathway activity during this nutrient upshift. However, metabolomic profiles alone, while the concentrations of metabolites do imply fluxes to some degree, they aren't always great for providing exact quantitative values of these fluxes. And so the next step of this investigation is basically, how can we quantify the relative use of the ED versus EMP pathway fluxes during these transitory periods? To do so, our lab hypothesized the use of asymmetric isotope tracers as a way to distinguish EMP and ED pathway fluxes. Now, an interesting feature that differs the EMP versus ED pathway fluxes, ED pathways, is that when you process a single molecule of glucose, for the EMP pathway, the initial hexose phosphate gets cleaved into 2 triose phosphates, which all fall down lower glycolysis. However, in the ED pathway, only 1/2 of the hexose phosphate is actually split and gone down into lower glycolysis, which has the intermediates of GAP, 3PG, PP, among others. The other half of that initial hexose phosphate is immediately shunted into pyruvate. And so essentially what happens is that you have this imbalance in labeling or contributions of different halves of glucose when you funnel them down through individual metabolic pathways. By using isotope tracers and liquid chromatography mass spectrometry, we're able to actually measure these individual metabolites at a time and get their mass to charge ratios and identify for a particular metabolite what proportion of them have the abundance of what proportion of them have certain isotopomers or different amounts of isotope tracers. And so thus, just looking at this initial simple model, if we use 1,2,13C as our example tracer of choice, we're able to kind of map out these general basic functional equations that can lead to insight into the ratios of ED versus EMP pathway fluxes. Now, in the bigger scheme of things, this simple model of just the EMP pathway and ED pathway isn't the perfect representation, but it's a good explanation of initially how we're going throughout our analyses. Realistically, to get a more accurate estimation of these fluxes, you incorporate the contribution of asymmetric glucose labeling across different pathways, such as the pentose phosphate pathway. But essentially, the goal here is that we can use the mass isotopomer distributions of single lower glycolytic intermediates and develop some sort of not too complicated mathematical function that relates the ED to EMP pathway ratios to each other. So to first test this hypothesis, we conducted this analysis on purely steady state growing cells, which don't have dynamic does not have dynamic metabolic changes. So we grew nutrient-replaced cells in 1,2,13C glucose as their carbon source, and then extracted metabolites and ran our samples through LCMS. And we did our corresponding flux calculations based off of the hypothesized equation and compared them to more rigorous flux analysis techniques, such as metabolic flux analysis, which instead rely on the metabolite labeling profiles of a whole span and a larger system of metabolites, and is much more computationally intense. And the good news is that the results of these studies is that we find our estimations of the ED to EMP pathway fluxes based off a single metabolite's MID to be roughly similar to more rigorous estimations in the past. And so this gives us the confidence to use this kind of experimental approach to study metabolism in dynamic systems. So going back to the original growth nutrient upshift experiments, we conducted a very similar setup for wild type cells growing inside these systems, except now we're feeding 1,2,13C glucose as our tracer, so we can get a larger amount of information when we extract our metabolites over time and run them through LCMS. And so the results of this study, I tried to plot them in this what I call kind of like a walk through metabolism. And so the best way to kind of understand and read this heat map here is that we can solve the ED pathway, ED to EMP pathway fluxes and their ratio, which is just the color scale on the right bar, based off of a function of just the unlabeled 3-phosphoglycerate or 3PG percentage on the x-axis and the singly labeled 3-phosphoglycerate percentage on the y-axis. And then we can plot these individual points of labeling at different times after our nutrient upshift. What we can see in the case of glucose upshift initially is that we can see that during the gluconeogenic growth stage, ED pathway usage is initially quite low in the glucose limited state. However, immediately after nutrient upshift, the ED pathway usage increases as it walks over towards this darker blue region until it reaches a value that's more akin to the steady state values in our previous experiments. Another unique feature that we noticed by mapping it from this kind of biochemical walk instead of just individual ED pathway fluxes over time is that when you compare it to the nitrogen upshift condition, the start and ending trends of ED pathway fluxes are similar in that the ED pathway fluxes relative to the EMP pathway are also initially low in nitrogen limited state and then reach a steady state value after upshift. But the interesting thing that you can kind of get from this graph is that in the case of nitrogen upshift, the ED pathway usage increase follows kind of a different path of labeling profiles between glucose and nitrogen upshift. And so this kind of provides some more unique insights on the different mechanistic ways that metabolism is altering itself in response to these different types of nutrient upshifts. And we also collected these data as a whole to kind of map out some general trends we can use from this data, and that we can kind of come to the conclusion that the general labeling of lower glycolytic intermediates when using an asymmetric isotope tracer, such as 1,2,13C glucose, can reflect general characteristics of cellular state. And while these trends don't have exact quantitative relationships, the general trends represented on this figure are kind of a good way to take snapshots of metabolism over time when you don't want to conduct more rigorous and kind of holistic metabolic flux analyses. So kind of as a summary and outlook of everything that we've learned today is that we first kind of done a computational kind of estimates of steady state growth for fluxes for the ED pathway and ED knockouts. And we demonstrated that the ED pathway seems to have a minimal effect on steady state growth. But when we investigated nutrient upshift conditions, we see that the ED pathway actually influences the rate of transition between these growth phases. And to kind of get a more quantitative analysis on how these fluxes change over time, we're able to map these biochemical walks of how metabolism is changing from a low nutrient to a high nutrient state. And hopefully through all of these results, we come to some sort of conclusion that shows us that the ED pathway helps enhance equalizability to rapidly increase pathway fluxes. And in kind of expanding this broader to beyond just the scope of microorganisms, this kind of hints at there's more metabolic features to parallel pathways than just, say, the idea of metabolic redundancy in the case of single gene knockouts rendering a cell to be completely lethal, right? Which is the case of synthetic lethalities. Instead, there could actually be more biological phenotype responses that are kind of a consequence of possessing parallel metabolic pathways. So I just want to take this time now to thank everyone, the members of my Park Lab, specifically my advisor, Dr. Jun Park. And obviously, I want to thank all of you guys for listening to my talk, and I'd be happy to take questions.